Richard Gilbertson, who'll be talking about pediatric brain tumours. Thank you, Richard.
Thank you very much. Do I just plug this into here?
Let's assume yes.
I hope so. Ooh, yeah, looks like something's happening. Yeah, great. Well, thank you so much. Thanks for the invitation. It's great to be here. And a few disclosures up front. I'm not a mathematician, nor am I a computational biologist. And I'm also conscious that I'm between you and your sort of early afternoon nap. So, in fact, I said that at a meeting recently, and somebody said, not necessarily. So, anyway, we'll see how this goes. But as Steve just said, I'm actually originally a children's cancer doctor, although I don't see patients anymore. I spend all my time completely running the lab. But I thought I would take you back to what it's like to have a brain tumour as a child as a starting point before I get into the computational side. And what my lab's done for shockingly 30 years now is to identify the origins of children's cancer. Let's see if I can get a pointer on here. So to identify the origins of children's cancer. So when I first started looking at kiddies' brain tumours, they were basically treated all the same, roughly diagnosed down the microscope. There was no genomics or anything. And over the last sort of 20, 30 years, we've noticed that children's brain tumours can be divided into different genomic subtypes, like many other cancers. But what my lab was able to do is demonstrate that actually those transcriptomes are not abnormal, but they actually reflect normal lineages in the developing embryonic brain. And in doing so, we've been able to work out the origin of a lot of brain tumours in kiddies. However, like all science, all we ever did is replicate what had been done 100 years earlier by these two amazing people, Harvey Cushing and Percival Bailey, a surgeon and I don't know what that is, a surgeon and a neuropathologist. And nothing but a microscope and the back of an envelope, they drew all this out, which is pretty incredible, really. But I think what it's worth thinking about before we look at some of the processes that we've approached in the last 10 years or so is we've reached for computational approaches, and a number of people have said this today because obviously we're drowning in data, we all know that. Computational approaches can actually help us with this. But actually what we do to children is incredibly complicated. So if you're a child who's diagnosed with a brain tumour, you'll be about somewhere between 5 and 12 years old typically. You'll go through imaging, you'll have your tumour resected, it will then be staged, in this case it's medulloblastoma. You'll almost invariably get radiation and chemotherapy. Now about 80% of you will survive. However, after surgery, about 20% of those kids will get something called posterior fossa syndrome, which means when they wake up, they never speak. They lose their balance, and they never have long-term coordination anymore. On top of that, if we try and reduce the toxic effects of chemotherapy, about 90% of those children will ultimately relapse. So we know we need to give that sort of level. And chemotherapy lasts about a year out of your school when you're a 7-year-old. So this is really shit. It is appalling, actually. And we have to do that to children because we really don't know how to treat it. But it gets worse because of those 20% of children that drop off the end of this and actually relapse, we have no idea what to do. We pull a drug off the shelf, possibly because some protein is expressed in their disease and there's a drug used in adults, for example, a receptor or something like that. We give it to a child, typically about 10% of kids will respond in a clinical trial. We have no idea why they respond. They relapse and they go through this cycle again. And so obviously this is really, really not good. So we need to have a better way of understanding this. And some of the approaches that have been developed is the sort of rational combinations, but they aren't rational and they too often fail, and you know this. So we sort of stylize the way that biology works and these nodes and linear pathways at which drugs can intersect. And it's complete nonsense, because of course the system is totally fluid and doesn't work like that. And you've seen that through some of the beautiful talks today, even just modeling these things. The system is far too complex, we have an inadequate view of cancer evolution, and we fail to predict efficacy and particularly resistance mechanisms. So along comes single cell sequencing, and you know, many of us will be old enough to remember when it first started. I remember when I first got my Affymetrix array data when I was in the States about 25 years ago. It was a pile of paper that was literally 6 foot tall. What I was supposed to do with that, I have no idea. There was no such thing as computational analysis for that. And so what we thought single cell sequencing might do is help us, but actually this is actually single cell sequencing data from a patient with a brain tumour. Now, the cell states don't necessarily easily separate, but I think this is a really, really important point, which I'm not sure has been brought out a lot today, and that is that we will look at the gene expression patterns in cells, and then we define what that cell is by looking at something in the literature. So we will look at for analogous or similar pathways. For example, there are progenitors that have been associated with this particular pathway, so we will call that cell a progenitor. But this is a totally different context. We might miss contexts that are relevant here, or actually prescribe contexts which are not important. Sometimes it's very easy to tell differences between cell types, but not others. And then how do we compare the static information between states? And we've heard a lot about that today, some really fantastic talks on that. So this is a paper from the lab. I'm just showing this cover because it's actually this month's cover of Cancer Cell. So this paper is in this, but the reason we've got this beautiful picture by Frank Dixie, who was a fantastic painter towards the end of the 1800s, beginning of the 1900s, of Romeo and Juliet, is because I think it's an analogy for the very simple approach that we've used. So that is that there are 884,000 words in the complete works of Shakespeare, but if I say balcony, you will all say Romeo and Juliet. And that's word embedding. It's simply that you have learnt that balcony only goes with Romeo and Juliet. It doesn't go with Henry V, whatever other play you want to choose. And so Amir Hasim, who was a postdoc in my lab, and he's now in Columbia in New York. But actually, he developed this approach, and actually this is a really nice summary from the preview that went with the paper, which positions what we call context drift somewhere up this continuum, beginning with differential expression analysis all the way up to multi-omic data integration. And I'm going to go through what gene context drift means. So this is a snapshot of some of the kinds of data that you've seen. It's single cell sequencing data. You know, it looks like the pavement outside a pub, as I say, where someone's come out and thrown up. But you know, how on earth do you pick out the information that's in there? And so what this does is quite simple. It simply takes the gene expression data from each cell, and then it looks at the ranking of genes in each of those, and then builds what's a very simple approach to what's been used today. Is it builds a graph of similarities of gene expression across that entire network. Now the difference is it's completely agnostic to what those genes are. So imagine we have a simple network here, which is, you know, say before treatment, and you've got these genes and they form two different communities, and they're related to one another by how much they correlate across, say, 20 or 30,000 cells. Now, if you build a network after, say, treatment, or if that was a cell of origin and this is now the cancer, the graph has evolved. Now, the bottom part of the graph has stayed pretty much intact, but the top part of the graph has changed quite a lot. And so what this does is uses 4 simple rules to quantify for every single gene on a graph how that has changed. So it uses something called a number score for the neighbor, graph for each neighbor cosine score, and basically, I won't bother going through each of these, but it's a bit like looking at the constellations in the sky, plotting where a star is in, say it's in Cassiopeia, and then the next night it's in Orion, it's moved. And it quantifies where that particular gene is in the galaxy of the transcriptome relative to all other genes in the transcriptome. And that's it. It's pretty simple. But what it allows you to do is then ask, what is the transcriptional state in the beginning of that treatment to afterwards relative to the entire transcriptome? So what is what we call context drift? I'm going to use 2 examples to show that. The first one is published, the next one is not at all published, and it's just a bunch of data to show you. And we chose a very difficult disease, chorioplexus carcinoma. This was the first disease that I ever treated. Just as a side here, just to put this in context, these were 2 parents who had gone through 4 rounds of IVF. They were in their late 40s, they'd never had a child. They had a child, was born with a brain tumour, died within 2 weeks. Absolutely horrific. And this child had chorioplexus carcinoma, which is this big, horrible lump in the middle of the head. There are no clinical trials, there are no new treatments, it's too rare to study. About 2 children in this country a year get it, and the most likely outcome is death. So it's one of those intractable diseases where cancer's thrown us down a blind alley as opposed to us being able to put it down a blind alley. So what we did is, my lab accidentally made a model of this many years ago, and so we know which cell type in the nervous system it comes from. It actually comes from a progenitor at the root of the choroid plexus, and that transforms into this tumor type. And you're looking at very simple, just graph networks of genes in the normal choroid plexus in the embryo, and then in the tumor in the mouse, and you can sort of see groups of genes, progenitor, immune genes, epithelial genes, and how they are different between the sort of cell of origin and the tumor state. And you can start to use very simple alluvial plots to look at how these genes change their affinity with different communities over time. But much more importantly is the ability to track how these genes change their transcriptome context, this context drift, that very simple context. Now, the reason why this was a staggering result for me is that the genes that changed their context the most hadn't changed their expression level at all. They were expressed at exactly the same level in the primary origin as they were in the tumor. And in the traditional way of picking drug targets, we would never pick those as drug targets. The way we pick drug targets at the moment is we look for those genes that have been massively upregulated and assume they're important targets. We know that's complete nonsense. It often doesn't work. And it predicted that those genes involved in DNA repair will be particularly important. And indeed, we did a large-scale DNA repair analysis by comparing knockout of individual DNA repair genes. This is just showing a number of them here. The pink cells are those from this tumor. There's another tumor type and the cell of origin here. And in fact, indeed, by knocking out a large number of DNA repair enzymes, including one that was the most predicted by this recoder process as being important in the disease process, actually were critical for the cells to stay alive in organoid systems. So we actually inhibited that with a drug called AZ1390, which is an ATM inhibitor which gets across the blood-brain barrier. And indeed, what was remarkable is that whereas radiotherapy, which we know is effective against this disease, worked, for those who are not used to looking at these are waterfall plots. All it means is the bigger the negative result, the bigger the effect the drug has had relative to normal untreated control tumours. So radiotherapy of different schedules has a significant effect. But this drug, just by blocking DNA repair, as predicted, had an effect on its own. And it's still, to our knowledge, the only example of this DNA repair enzyme, or any DNA repair inhibitor, as a monotherapy having an effect against some form of cancer. However, that's not very interesting because you could predict that DNA repair might be important in this disease, and also it still doesn't help us with the uncertain cycle of clinical trials. So could we use this approach to predict if a tumour relapsed, how it was relapsing? Because at the moment all we can do is compare gene expression profiles before and after. So what we did is we took tumour cells before treatment and then tumour cells after relapse and put them through this machine learning natural language inspired process and asked which genes changed their context the most. And this was another surprise to us because this is the graph, very simple co-expression graph network, untreated. And then this is the graph networks of relapsed tumours. Now the top shows you the graph networks relapsed from a DNA repair inhibitor and the bottom from radiation. These are two completely different treatments, but they relapse in almost exactly the same mechanism, according to this. And so I'm just showing you one example of this. This is showing an alluvial plot as you go from the cell of origin. This is all in vivo data from single cell sequencing through the formation of the virgin tumour, then treatment and relapse. And you can see how the transcriptome evolves. One thing I'm going to point out, and I'll point it out later as well, is that typically what we would do is take an untreated tumour and compare it to a relapsed tumour. And as you can see, these numbers represent the number of genes in each community. There are more genes here. There are new genes acquired relative to the untreated tumour. We would assume this was new biology, something that the tumour was doing in response to resistance. However, what the alluvial plot tells us is almost all of these genes have been re-remembered. They're basically functions that existed in the primordial cell of origin in the brain and have been reactivated in a resistant tumour. So you're starting to see, as we've seen in other disease types, how these sort of repair mechanisms, if you like, cancer being a damaging process along with treatment, are being reactivated. Now, because this gives you a score which is a continuum, you can actually look at, and you can, for each gene, look at the ranking of those which have associated treatments, where they appear, and it's telling you in terms of context which genes to the left are the most important in driving relapse. None of these genes change their expression. They simply change the partners that they work with in terms of the tumour after relapse. It predicted that actually part 1 would be an important driver of this, and so we devised a number of clinical trials. And again, when we added part 1 to this either monotherapy of AZ1390 radiation, significant increase in response. What was important is if you give part 1 inhibitors to an untreated tumour, it's completely ineffective. So it only works in the context of relapse as predicted by this particular mechanism. But again, we don't give patients monotherapy, we give them combination treatment. So can you do this in the context of combination therapy? So we did a very large series of preclinical trials in which we did every single combination you can imagine of the DNA repair inhibitor AZ1390 and radiation, treated many hundreds of mice using this particular protocol, and repeated this recoder analysis. It was a huge amount of work. And this was the third surprise. So here's the untreated graph. Monotherapy relapse, as I showed using either treatment, have very similar relapse. And you might predict, if you're a Dumbo like me, that actually if you add these drugs together, if they relapse in a similar mechanism when they're given in isolation, then actually if you combine them, they'd still relapse with the same mechanism. Absolutely not. They relapse with a completely different mechanism. And this is a really important message for when we do combination treatment. We think that we can understand monotherapy relapse, but actually when you put those together, all bets are off. And when you put two combinations of treatments together, there's a completely different pattern of biology that emerges. And all this is really doing in an agnostic way, without forcing any prior knowledge, is telling us to look at how these tumours are relapsing. So again, we can build this sort of history going from the cell of origin in the embryo of the mouse, through formation of tumours, through mono-relapse, all the way to combination relapse. And once again, you can see these re-remembered pathways. They were very heavily deployed in utero and have now been reactivated in the relapsed tumour. And actually what happens here with the emergence of a large amount of immune cells, we've heard a lot of talk about immune cells today, and I caution people to assume immune cells in tumours are immune cells. And so this is a well-known phenomenon called myeloid mimicry, which has been well described in the brain, and like many people do, Steve Pollard's described this in glioblastoma. And actually when we combined DNA repair inhibitors radiation, there was this huge upsurge in TREM2 positive cells. Now the advantage we have in our tumours is they're GFP positive. So most of these TREM2 positive cells, which marks macrophages or microglia, are actually tumour cells. And we could prove that because they all have abnormal genomes when we do concurrent FISH. So actually what was happening is the transcriptomes of these cells were indistinguishable from tissue-resident macrophages, but they're tumour cells. And they're mimicking this resident tissue type, this resident cell type. So we again did this analysis where we basically ranked genes in terms of their importance, both pre and following combination relapse. And strikingly, there was a single drug which has a number of targets called dasatinib, which is used basically to treat leukemia, would never be really used to treat brain tumours, which came out really at the top, and it targets many of the genes that were showing significant context drift from the before treatment through to combination relapse. Again, none of these genes, apart from one actually, were differentially expressed between the primary and the secondary tumour. They just formed new neighbours and new networks. So again, we designed a series of quite detailed preclinical studies in which we added either dasatinib alone, or we gave dasatinib in combination with the monotherapy AZ1390, or different combinations of radiation. And this is when we saw the most dramatic responses, and particularly important, the addition of dasatinib after combination treatment meant that many of these tumours didn't relapse. The mice died of other causes. So actually this is now going to be the first international molecular targeted therapy in these children with cryoplex carcinoma, which we're going to be doing in concert with AstraZeneca and with Boston Children's Hospital. So does it work in patients? So this is my last slide, is the first part of the talk. And what you're looking at here is sort of classic unsupervised hierarchical clustering, but this is not gene expression. None of these genes are differentially expressed between patients. What we've done here is to use two very remarkable data sets that were collected by Michael Taylor, who's at Texas Children's Hospital, who was a postdoc in my lab, has been an independent faculty member for a number of years, and from Jean Abraham. We've already seen Jean's work presented by Jasmine today, and we used 47 patients who had their single cell sequencing done before treatment and then after treatment. And what you're looking at here, both in the medulloblastoma patients and also in the triple negative breast cancer patients, is a hierarchical clustering of their gene context drift scores. So it's telling you how these patients line up based on the remodeling of their transcriptomes. And again, the genes that are dividing these patients up are not differentially expressed between these different groups. And what was striking to me was these 4,000 or so genes in this group B. So this group of women with triple negative breast cancer are indistinguishably molecularly and clinically from the other three groups, but they have this large amount of remodeling that's happening during treatment. And so this suggests that this might be a relatively unique group of women who are following now with genes to see if they've got a very plastic genome that's able to respond to initial treatment and thereby undergo a level of resistance. And we're hoping, of course, given the predictions that we get from this, we've got treatments that we can offer those women should they relapse. Okay, so in the last 4 hours or so, what I think I'll do as a joke is go through a second idea which we've been pursuing really now for about 15 years in my lab. And that is the concept whether development sets the ground state for all cancers. Now, we know children's cancer all arise in utero. We've shown that in brain tumours. Other groups have shown that in solid tumours. And it's quite easy to see that in children because in children, it doesn't matter where you look in the world, largely, leukemias are the commonest, followed by brain, followed by kidney, followed by muscle, followed by retinoblastoma. So there's a very prescribed order of tumour type in the incidence, both male and female predominance, and also in the tissues that they occur in, and they look like developing tissues. So there's no question that development is involved in children's cancer. The problem is we make the erroneous sort of a conclusion that somehow development ends when you get to vote and then development stops. That's complete nonsense. Development continues throughout life. We call it aging, but it is still development. And there are active processes in the body which are ongoing, which I'll go into, which govern this aging or continual development process all the way through to when you're 6 foot under. And we know that because actually, if you look at the incidence of cancers in adults, they're also prescribed. You can have a league table of the commonest incidences of cancer in both men and women, and with some exceptions, they're largely invariable across the world. Now that's not random. That is because it's governed by inherent processes within tissues. So it's worth pausing and thinking a little bit about theories around the cancer of origin. Now, the first theories theory one really, which was developed over 100 years ago, is the somatic mutation theory, which took off particularly with the discovery of, with genetics and then the discovery of the DNA code. And the concept here obviously is that oncogene activation, tumor suppressor activation, is the heart of everything in cancer. Now, of course, we know that DNA is important, but we know it's not the sole player in cancer. And so a second theory that came along was the tissue organizational theory. And that was recognizing that because mutations in themselves are not adequate enough to cause cancer, there must be some other aspects that are driving malignancy. And this related to tissue disruption, damage, and it allowed the concept that things like inflammation can accelerate cancer. The third one was the bad luck theory, which got so much bad press. So this came along, and you may remember that Bert Vogelstein and Christine Tomasetti proposed this in Science, and they made it worse by putting out a second Science paper, which was even worse. But basically, the concept here is that cancer is completely bad luck. Because it probably happens in stem cells, and because it requires mutations, and mutations are random, then it's just bad luck. It's going to happen. But actually, although there's clearly a truth, as there is truth in all of these theories, it can't be explained alone because there are certain epidemiological differences around the world which were marked variances in cancer, for instance, which can't be explained by bad luck, and they were sort of completely ignored by the hypothesis. So then, about 10 years ago, my lab put forward this ground state theory, which sort of unifies these theories. And that's the concept that actually, yes, mutations of course are important. Yes, extrinsic factors are important. But equally important is the ground state in which a mutation happens. There's something about the tissue in a neonate, in an adult, in a teenager, in a particular organ, and its epigenetic makeup and the holistic aspects of the cells around it, which are defining whether or not this cell is predisposed to making malignancy. And one of the ways we've explained it is like this. Now, 1 in 600 kiddies, fortunately only 1 in 600 kiddies get cancer, but 1 in 2 adults get cancer. And the dogma's been that you have to be around for, 3 decades, or 5 to 6 decades, slept with the wrong people, drunk the wrong stuff, smoked the wrong stuff to get enough mutations to get cancer. Right? That's the sort of theory that prevails. But of course, that's nonsense, and we know that's not true. And the dawn of beautiful work that's been done in the skin and in the colon and now in the blood and other tissues, we know that by the time you get to sort of my age, most of your tissues are mutant. Most of them are full of mutational clones, and yet you never see cancer in many of those people. So why is that happening? Why is it you can tolerate mutations? And so what we think might be happening in the neonate is that actually the natural process is lineage restriction and differentiation, recognized sort of by this downhill slope. And so that's the natural process. And actually what we hypothesized is that, and I'll show you that we've proved this, at least the concept of it in the next slide, is that actually to become a multi-organ species, we must have involved some mechanism when we were in utero and in the early years of life to stop us getting cancer. Kiddies are essentially proliferating balls of flesh with huge amount of differentiation stress. So there must be a mechanism that we've evolved to stop proliferating immature tissues from simply transforming, because mutations are fairly frequent. Now that could be DNA repair, it could be an immune surveillance system. It's agnostic to what the mechanism is. But when you get to an older state, you don't need that anymore because you've already passed your genes on evolutionary, etc. But you do need to repair. And so one of the reasons we think why mutations might be tolerated later in life is because, a bit like I used to have a really crap second-hand car and it was always needing to be fixed, is as you get towards the 4th, 5th, 6th decade of life, you actually rely on these mutations to keep your tissues going, to keep the proliferation going, to keep you able to divide. Now the payoff of that is that some of us, 1 in 2 of us, will get cancer as a consequence of that. So it's a slightly different way of looking at mutational spectra. Rather than being abnormal, is being a natural consequence of evolution to maintain tissues. Now, is there any evidence for that? Well, Li Chinzhou, who was a great postdoc of mine 10 years ago now, when I was at St. Jude, so she did this work. Li Chin now has her own lab. And we tested this by a quite simple but very laborious long experiment in which we took mice which we'd made that marked the prominent one on CD33 with a Cree RT2 cassette. If you're not familiar with that technology, don't worry. All it means is that you can activate a fluorescent marker in a cell and all of its progeny from then on will be fluorescent. And we activated that marker in babies, so kiddies at P1 mice, or in older middle-aged mice, P16. And then we aged them. And we looked at what those cells were contributing to the tissue in terms of proliferation and stemness. Now, in an identical parallel set of experiments, we used 9 different oncogenes or tumor suppressor genes to actually activate the kind of oncogenes or oncogenic effects we see in human cancer, to represent like the waterfront of human malignancies, and then age those mice. And what that allowed us to do was to look at one side, what is the normal fate of those cells throughout life in a neonate or an adult, and then what is the fate of those cells if you give them a mutation. So you could age mice looking at their cancer risk, and you could age mice looking at their normal stem cell function. And so there was some quite interesting difference. So like these PROM1 cells that exist around the portal system in the liver, less than 0.001% of all cells in the liver. If you lineage trace those in a neonate, the entire liver is fluorescent by the time you get to 3 months. It makes the whole bloody liver. It's absolutely amazing. However, within about 3 months, they become completely quiescent and they don't activate again unless you damage the liver. And it's that dynamism, that difference in cell function, which is really important to think about when we're thinking about origins of cancer. Now, this is probably the most important result. And so what we've done here, with the help of Arzu, who's a fantastic mathematician at St. Jude, is actually, she invented some new logic rank maths to do this, is we looked at lifelong lineage tracing. So this is across all organs in mice, so hundreds of mice. We looked at any given cell, it doesn't matter what organ they're in, what their lifelong normal stem cell function is, and then what their tumour probability is, their lifelong risk of making a tumour, if you introduce mutations into those cells. And what was interesting is that if you have no stem cell function, so Bert Vogelstein and Christian Tomasetti were right, actually, if you don't have stem cell function, you will never get malignancy. They're completely resistant. And actually, the more stem cell function you have, the more likely you are to make malignancy. But this was the striking result, that for a given amount of stem cell function, neonates, even though they carry those mutations for 60 days longer throughout life, are 7-fold less likely to make cancer than their adult counterparts. So this demonstrates that there is a protective mechanism that exists in neonatal tissues to stop you from getting cancer. So how can we get at that? So that was 10 years ago. So what I'm going to show you now is a bunch of data that Amir, before he left, has generated over the last 10 years. So the brain is appropriately over the brain. So what Amir did is he took those same 12 organs that we already had functional data from 10 years of lifelong lineage tracing in cancer, and then we've carefully isolated single cells from each organ systematically throughout the whole of aging. And we've generated transcriptomes with Rudi Ebersol, who's now retired, who was in Switzerland. That shows you how long the project took. We've developed the proteome for these cells, and with Wolf Wright, when he was at the university here before he went to Altos, the transcriptome. So we have a very large data set now of all of this information. So what you're looking at here is a single snapshot of all of these data on a single unified graph. And it's self-organizing, as I mentioned. We don't impose any information on this from outside. So it's self-organizing based simply on co-expression graph networks. And you can see real stuff. So yes, there's genes, so the cell cycle goes together, immunity goes together, etc. And then because this is, the approach is agnostic to platform, you can combine these so you can build graph networks, transcriptome, proteome, etc. And I think this is a really important piece of data. I've just animated this. What you're looking at is how these relationships change over all 12 organs throughout life. And this is an example of what I call the Cambridge professor. So this is a gene that basically got its undergraduate in Cambridge, it did its postgraduate in Cambridge, and its doctorate in Cambridge, becomes a lecturer in Cambridge, and it dies in Cambridge. It doesn't do anything else. It just stays in Cambridge throughout its entire life, and its colleagues are always the same. And then you've got other genes which are remarkable, and this is a really important point, because I've heard a lot of people say today, using PubMed, etc., and that's great. Obviously, that's our data source. But what was an interesting exercise we did with this is we linked PubMed into this. So we actually put 8 million references from PubMed into this graph. And what was striking is that at certain points, PubMed was correct. In other words, if sausage gene was discovered in skin cancer, in malignancy, and in all of the sort of bandwagon that sort of carried on from the community around that, and it discovered that P53 was involved in it, was a P53 regulator, and then P10 was involved in all that kind of stuff. At some point in time, that's all true. But for the rest of life, it's like this green gene here, which is just going off and doing something completely different. And so that agnostic approach is really important to bear in mind, that we don't get sort of dogmatized by the literature. So I'm just going to show you a few slides which show you, because we're just starting to analyze this. This is what the data looks like in terms of RNA-seq, whole genome bisulfite, proteome, etc. As you might expect, this data clusters quite nicely by organ site. So this is on the left, we've got the whole genome bisulfite sequencing, proteome, and transcriptome. But just some initial analysis that we've done, obviously following the hallmarks of cancer, there's now the hallmarks of aging. And you can see, if you line up all of the genes across all organs over development, some of the genes do obey the literature in terms of what's predicted to happen with the hallmarks of aging, but some of them don't. And this is the sort of worry I have about sort of these hallmarks that are appearing, is that actually when you look across all tissues, the effect has disappeared, and it's very idiosyncratic to a single tissue or to a single state. And that's really important to bear in mind when you look at this data. However, you look agnostically, you do see remarkable waves of biology happening. So each of these is a methylome, transcriptome, and protein. And this is over periods of time from birth all the way through to old age. And it's just representing how you see changes in various cell functions over time. So obviously we're mining this at the moment. So what's the sort of big idea? Well, the big idea is we've built 213 graphs now. And we've released agents onto those graphs. You've heard that phrase already today. And they've now completed just over 100 million walks across all of these graphs. And so what we've done now is taken those graphs and they learned the context, just like they know balcony goes with Romeo and Juliet, which is where we started. So across all of these graphs, they've taken those walks. And this is just one representation of what it does. So what we've been able to do now is, so for example, if I said to ChatGPT, write me a poem about roses, it would write me a beautiful poem about roses. In an analogous way, what we've done is we've taken some of the training walks, which usually cost a system about 60 genes, and we've halved those prompts, and then put those halved prompts back in, and asked the machine to finish off the rest of the sentence. What's really cool is many of those graphs have not seen proteins, methylome, or transcriptome together, but the machine puts them together. It says, I think the next thing should be a protein. I think the next thing should be a methylated region. So it's finishing off these sentences. And if you give it a prompt from the cell cycle, or if you give it a prompt from DNA repair, it absolutely faithfully finishes that off with the cell cycle or DNA repair. So what we're doing now, and this is just a snapshot of this, is going back to why we originally did this, is what is it that a neonate has that an adult doesn't? So we have these graphs in the context of damage. So what we're doing is providing damage prompts in the adult to the neonate and comparing how those sentences are finished off in the neonate and how they differ from those that are finished off either in cancer or in an adult. And it's those differences that we think will unmask what is this protective mechanism, if it indeed exists, in the neonate. And the obvious reason for that is because obviously if we can reactivate those kind of genes in adults, we may be able to develop some kind of statin for cancer to prevent cancer. And what you're seeing here is just some of those shifts that happen in one sentence. It's just represented by this graph. And these blue dots and half sort of moon dots are where it's introduced genes that are, it is not previously seen in the same graph. So it's picking out from the 100 million walks alternatives that it hasn't seen before together, but it's suggesting actually are the best fit. This is my last couple of slides. So we're actually been trying for the last 3 years now to build a virtual child. There's a great comment earlier on about how, absolutely correctly, these are going to be built in single systems. And so what we're doing at the moment is building the nervous system using chick, human, and mouse embryonic and large data sets. And we're actually building a nervous system which will generate tumours pretty well, you've seen, but we'd like to recruit teams in the blood, in the liver, in the lung, in the muscles for them to do this, and then eventually bolt them together into some kind of system that will create a true virtual child. And the reason for that is obvious. One is that, obviously you can start to think about the sort of computational virtual trials we've talked about today, but also for drug companies in terms of on-target, off-tissue effects. So it costs billions to do clinical trials, and of course many of those fail because of toxicity. So if you had at your disposal an aging human, and you could predict where a drug might affect at certain points, that might also be very helpful in terms of looking at therapeutic development. And the team here, which is my last slide, are working together to do this. If anyone's interested in joining us, we're very welcome. And this is my lab, and thank you very much for your attention.
Are there any questions people have? Yes.
So I'm going to begin this question using the kind of mathematician's phrase that always winds up physicists. If one assumes that cancer is a disease of immune evasion, what I'm thinking of is the point you made about the number of errors that you have in adults compared to children, and that ability to deal with all of those mutations, is that in any way connected to the maturity of the immune system?
So that's a really good question. So actually we have a paper in revision at the moment. So one of the questions I've always had, is a really good question, is why do children not reject brain tumours, or why do they not reject sarcomas, for example, muscle tumours? And we haven't, previously the brain was thought to be an immune privileged site. There was no, it was dead to the immune system. It was behind a castle wall and the immune cells never got in it. We know that's nonsense now. And actually, Elizabeth Cooper in my lab has shown that actually the reason why it happens is because brain tumours evolve with the normal brain. And they simply hook onto the system which recognises brain as self, and it educates the tumour as self. Now what's amazing, she's worked out that circuit, and an infusion of a single antibody which breaks that circuit, the tumours go away, they get flooded with T cells, and basically those cells will basically remove the brain tumour quite effectively. They exhaust and the tumours come back, which you can get rid of with checkpoint inhibitors. The reason for mentioning it is I don't see why there shouldn't be anything similar in adults. And actually, you know, before we have this paper, there was a beautiful paper about a hemopoietic antigen-presenting cell in the bone marrow, very similar cell type, which works in leukemia. I think we're going to find this interplay between understanding and recognizing what is self and how cancers actually utilize that process to prevent them being rejected. So we have this sort of, I think, personally misunderstanding that cancers somehow magically are able to cloak themselves in the immune system. Well, actually, all our tissues are doing that all the time. You know, why aren't we rejecting the colon, which is full of mutations? Why aren't we rejecting the skin, which is full of mutations? Well, it's partly because the immune system is constantly being educated about what is normal and what is abnormal. If we put a kidney in you, as we saw in the last talk, you know, you would immediately reject it. So I think there is lessons to be learned from normality, that tumours aren't as grossly abnormal as we think. They're just using systems that exist in normality. I think taking that on board will be critically important for the models that we've talked about today as well. Sorry, that was a long answer.
Yeah, very interesting. If I understood correctly, your kind of analysis of how context changes, you kind of restricted it to kind of individual organ systems kind of through time. Is that right? You've looked at lots of them, but you kind of thought about the liver or the kidney.
No, it's a great question, Eric. So, yeah. Yes. Yeah. So obviously no time to explain it all, but so exactly right. So what we've been looking for is, are there common mechanisms in place that are susceptible states for cancer? And one of the reasons for doing the big experiment first, which we published 10 years ago, is that we now know which cancers have the highest risk, intermediate risk, and low risk. So actually we can feed that information into this holistic view of the organism and seeing how do these graphical changes correlate or relate to different cancer risk across different organs. And so actually that question is critical. We've not done the liver and the muscle, and we're actually looking holistically, but using what we've known functionally from lineage tracing and also from cancer risk to try and feed into that.
Do you think, I'm interested in the liver, could you unpick how what's going on in the liver is impacting on how wiring is happening in other organs?
So I think Eric's question is a good one. It's about, you know, inter-organ communication, if you like, in real time. So the only data we've got which might help us with that is that we also damaged the liver, and we also damaged the lung, and we damaged the colon as a mechanism to accelerate malignancy, because it accelerates stem cell function. But we collected the cells from other organs at the same time. So actually, can we look at comparative age states in those relative to the damaged states in organs that weren't damaged, if you like? So we could look at that, yeah.
Sure.
It's very thought-provoking, and if I understood right, the main data is about the genes which are correlated in their expression and how those correlations change through aging. And then from that, you infer that a particular gene might have some different function because it has different partners later in life.
Broadly, yes.
But is it actually, are you just seeing an evolving epigenome where different regulatory regions are silenced or not, and different genes are silenced or not, and that's actually the mechanism that's going on?
So we're completely agnostic to the underlying mechanism. What we're looking at is simply how the genes change in the latter information, their relationship in terms of proteome, transcriptome, and methylome. Now, we can answer that question because we can actually build on the graph methylation changes. So what is interesting is you'll see individual genes changing a methylation state, and a whole slew of genes which aren't in that methylated region changing in concert. And because the data is so large and powerful, therefore you can make those correlative predictions. But it's correlation. It's a correlation network at the end of the day. To my mind, it's just a way of trying to encompass a level of granularity of the data and the biology in a way that is not fettered by existing knowledge. It's completely agnostic. So we're just looking for patterns and changes. Now, in the first part, it does so happen that those genes that change their context the most happen to be very good targets. So we've done 3 of those now in vivo, and we've done them in large-scale in vivo studies. They take a long time. But,
yeah. Is your opinion that the ultimate driver is the epigenome, or is there something else that controls?
I mean, it may well be the epigenome because that's an obvious way of modelling. What we haven't done is, so what we've been asked to do, and I think will be very difficult to do, is we do know that, for example, copy number changes happen as tumours evolve. So some of the change might be driven by copy number changes, etc. It could be epigenetic changes. I think the other thing that's complicated here, which is why I used the example of myeloid mimicry, is the cell populations are changing. So it could well be nothing necessarily to do with epigenetics, but there's an outgrowth of a clone that has a completely different transcriptome. So it's not that you've got cell type A that stays as cell type A but now has a different epigenome. Cell type A was 20% and now it's 1%, cell type B is outgrown. So to some extent it's a way of cutting across the noise of that, not getting at the mechanism, but trying to understand what the important transcriptome is as a corpus of text, which is the story of that particular point in the disease.
Last question for Hannah.
Thank you very much. Very interesting and provoking talk. You know, you mentioned this is sort of agnostic, the mechanism. But the next question, of course, is, therapies, treatment, and so on. Do you envision that in immediate future, in the next few years, one would have ability to look at this for, say, treatment-specific cases, or when you actually change conditions?
Yeah, so the patient data, so that group B of triple-negative breast cancer, or that group 3 patients who, so what's interesting, I didn't have time, but in the medulloblastoma group, they cluster exactly the same as transcriptome clusters patients, but those genes aren't differentially expressed. But it's still recognizing group 3, which is the worst actors, and they had the biggest amount of transcriptome remodeling. And the RECODA score tells you which genes have changed the most, and whether they have drug targets, because we linked it with the CANSAR database, which was developed by the ICR. So it does actually spit out for you not just which genes are changing the most, but what are the potential targets. So actually, Gene has that information, and we'll follow those patients to look for potential therapeutic intervention.
The question, how long do you need to follow?
Well, those women, it was the neoadjuvant therapy. Yeah, so it was before and after, which is a matter of weeks they get neoadjuvant therapy, yeah. And so what we're doing at the moment, we have a mouse resource with PDXs of triple-negative breast cancer, and we're seeing if we can replicate in mice what we see in the women, because we've got avatars, PDX avatars of the women. So can we see that in vivo, and then can we predict whether those actually are therapeutic targets in remodeling? Great.
Fascinating. Thank you.
Thank you,
Cheers.
Richard. So, we can thank Richard once again.